Couple of quick announcements today. One is a shameless plug for our article, which was just chosen as a spotlight feature in Journal Virology. So that's kind of cool. Second thing is clicker points. I should have uploaded all of the clicker scores as of this morning about 10 minutes ago. So please make sure that your clicker score is actually being counted. There's one person who's here who's clicking away, and I don't have their name. So there are a bunch of points there right now. The overall top score is about 75% on clickers, so that's about where we're going to be normalizing to about 30, and now it's about 38 points, but it's probably going to be a little bit lower than that. So just to give you a bit of an idea, today will be the last set of clicker questions, so after this I'll upload all the total clicker scores as well. Friday will be a review session. Final exam, when? Wednesday, yes. So at our normal time, normal place, normal exam, 50 questions. How many of you filled out your evaluations already for the course? Thank you. Those of you who haven't, speak now or forever hold your peace, or something like that. So today we'll continue talking about methods. This time it's all about the nucleic acids. Couple of things here as far as general concepts. The first one is recombinant DNA, why I brought my wonderful model here. It's really amazing that all of us and all of our bacteria and all the archaea in our gut are all made of the same stuff. And so that you can mix and match DNA is really, really powerful in terms of how to be able to think about understanding a lot about how we work, how bacteria work, how archaea work, etc. So we'll talk about that. The whole idea of recombinant DNA is just two different organisms, their DNAs coming together. One of the big problems that we have with DNA, it's wonderful if you've got a molecule like this, but of course this is way, way, way bigger than the real thing, and so is detecting your DNA. And because we've got this wonderful double-stranded thing that we've talked about before, you can pull strands apart and put them back together. That's a great way to detect a specific piece of DNA, or for that matter, RNA. But to do that, you have to have a way of detecting that little tiny piece that's come together into the right sequence. And so that's the whole idea of finding probes, a way to actually detect that specific sequence. And we'll talk about that. Blots, we talked a little bit about last time. We talked about identifying individual proteins on, in that case, it was a two-dimensional gel electrophoresis. We'll talk a lot more about the other kinds of blots today. Cloning, this is not cloning like Dolly, like the sheep. It's not a whole organism. Now we're just talking about cloning small pieces of DNA. And the basic idea there is you get many, many copies of exactly the same piece of DNA. And that makes everyone's life much, much easier. Then, where's that DNA coming from? Yes, you can go into a single organism and pull out one particular piece of DNA, but that doesn't do you terribly much good. You need to have many copies of it. You could make many copies of all of your own DNA, but people have done this already. These are the libraries, and literally it's a place you can go, or usually online, and say, hey, I want this piece of DNA from this particular chromosome in humans. And then someone will send it to you in FedEx the next day. DNA sequencing, this is really where technology has completely taken off in the last probably decade or two. But even just literally every couple of years, there's a new technology that comes online. And hopefully we'll get there. But the latest technology, it is able to sequence, if you believe the company, 6 terabase pairs of DNA in 2 days. Now, terabase is, of course, 10 to the 12. How many base pairs do we have in our genome? About 3 billion or more. So yeah, talk about sequencing. And again, this has been the real major change which has happened in DNA technology in the last couple of years. And we'll talk a little bit about PCR. Actually, PCR should have been a little bit earlier on in the whole process here. So why should we care about it? a lot of it has to do with the human genome and that sequence, which was literally just determined in the last few years. And now, if you've got 6 terabase pairs that you can sequence in 2 days, that means you can sequence hundreds of human genomes, literally in just a couple of days. In our lifetimes, your genome is going to be available to your medical providers and everyone else. The question is, should it be available to your employers? In fact, there was just a bill that went through the House that said employers can require having your genetic information being available. So, something to very seriously think about. Genetic modification, CRISPRs, CRISPR-Cas systems, etc. So, genetic modifications, we talked about CRISPR-Cas a couple of lectures ago. The whole idea there is we could potentially modify the genome in that process, and modifying of our own genome. People are talking about using human genome modification. What can you use as far as that's concerned? Drug production, how do we make a lot of drugs? What's that process? And the whole recombinant DNA technology process is trying to take care of that. Biotechnology, all of biotechnology is really based on all of the techniques, the recombinant DNA techniques that we'll be talking about today. Our research lab, plug for the recombinant DNA lab next term, the advanced molecular and cell biology techniques that we'll also be talking a lot about these recombinant DNA techniques. Last time we talked about protein analyses, all the protein analyses, oops, pardon me, down here. Here, again, we talked about analysis of our protein, determining amino acid sequences, or determining what that is as far as mass spectroscopy is concerned. You can then look in your databases for gene sequences. With those gene sequences, you can figure out what that DNA would be, encoding that. Encoding those sequences, you can then clone that DNA, try and make more of the protein through all the techniques that we're talking about down on the bottom here. The biochemical tests, these are the assays that we're talking about in terms of looking at individual fractions, and X-ray NMR analyses to try and determine your three-dimensional structure, which will give you a bit of an idea how things are functioning. So what we talk about, start out with gel electrophoresis. Again, a lot like what we talked about last time for proteins. Nucleic acids, it turns out, are a lot easier to work with. Probes in terms of doing detection. And again, this is hybridization analysis. Blots, where you put your nucleic acid or your proteins to detect them with all of your probes. How we make recombinant DNA has all to do with studying bacterial viruses, virology next term. I know 65 of you signed up for it. Restriction endonucleases are basically the other mechanism that we know of in terms of protecting a bacterial cell from infection by a virus. These, again, just like CRISPR-Cas, have been remodified and remodeled in order to use them for our own purposes. CRISPR-Cas normally is not present in eukaryotic cells. Everyone's using it for genome modification these days. Restriction endonucleases were critical for allowing the whole process of recombinant DNA in the first place. Vectors are what you have to have in order to make many copies of your favorite DNA. Usually these are small circular pieces of DNA that are good at replicating. We'll talk more about those vectors. Libraries, again, it's where you can get or purchase all of these different pieces of DNA from one particular environment. Polymerase chain reaction, making lots of copies. Anybody heard about PCR? You all know about PCR? We could explain PCR on an exam. Some of us. Okay, we'll talk a little bit more about it. And then DNA sequencing, bioinformatics we talked about already. So let's start with restriction endonucleases. Restriction endonucleases are how we all used to think that bacteria dealt with DNA viruses because they restricted the infection of various different viruses into a cell. How did they restrict the infection? They chopped up the DNA of those DNA viruses that are coming inside the cell. It turns out that they all have very specific, well, most of them have very specific DNA sequences that they bind to and cut. This is just the example of everyone's favorite. This is the restriction endonuclease from E. coli, the first one that they detected. Binds to and cuts a palindromic sequence for the most part. What's a palindromic sequence? The palindromic sequence is you have the same sequence on either strand. So in this case, GAA-TTC, GAA-TTC on the opposite strand. One obvious problem that you have with this wonderful mechanism, yes, the virus comes inside the cell, it gets chopped, its DNA gets chopped into little pieces, it can't do its job anymore. Well, unfortunately, your genome's made up of DNA as well. So how does that get dealt with? Well, the way it gets dealt with is DNA methylation. So all of the bacterial genomes that have these restriction endonucleases, and again, endonuclease means cutting in the middle of your DNA rather than exos from the end, they have to protect their DNA. The way they protect their DNA is by methylation. These are two of the common kinds of methylation here. I'm not expecting you to remember them, but that they're methylating their own DNA. And not surprisingly, if you look at genomes, usually you find a restriction endonuclease and right next to it in a genome a methylase, which is going to methylate all of the host DNA so that it doesn't get chopped up, but any foreign DNA that comes in will get chopped up. Turns out that there are many, 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 many of these restriction endonucleases. Again, most of them have these palindromic sequences. So we just talked about GAA-TTC. In fact, the very first one to be found was from Haemophilus influenza, HIN, Haemophilus influenza, AAG-CTT, and again, AAG-CTT in the process here. PST1, CTG, CAG. So again, these are identical sequences on the opposite strands relative to each other. There are a couple of things that can happen, and unfortunately the HPA1 site up here is cut off. Pun definitely not intended here. GTT-AAC, this restriction endonuclease cuts both strands in exactly the same place. So you end up with what's called a blunt end with that because there's no overhanging sequence. The other ones that we have examples for here, when you cut, you'll end up with either a 3' end, which is unpaired, or a 5' end, which is unpaired. And these turn out to be extremely useful because we also call them sticky ends. And so if you've got one of these overlapping pieces, then it can bind to your sequence, particularly of your favorite other piece of DNA, and they can come together. If you're interested in more of these, there's a place called Rebase at New England Biolabs. I think they've got about 2,000 of these different sequences available so that you can work with them. So this is how you can cut up your DNA. How do you know that you've cut up your DNA? You need to look at how they've separated relative to sizes. And there are really 3 different kinds of agarose gel electrophoresis to talk about here. Same idea as with proteins as we talked about last time. You've got anodes and cathodes, negative charge to positive charge. It's an electric field. Negative charges are going to repulse each other. What kind of charge do nucleic acids have? Negative charges. So you put the 2 negatives together and they will separate. As long as you've got a matrix with holes in it, those will then separate. The smaller pieces will go faster. The larger pieces will go slower. Depending on the matrix that you have, the holes are going to be smaller or bigger. So really small holes like this one here, the acrylamide, oh, it's interesting, can separate your DNA one base at a time. So the resolution here is such that this band right here represents a nucleotide, stretch of nucleotides, which, let's see, would be 16 nucleotides long. This band over here, 17. That one, 18. So it's got a resolution of single base pairs here. So you can literally separate something which is 10 nucleotides long from 11, from 12 to 13, etc. This is great, except that most of the pieces we're talking about, again, the human genome is literally billions of base pairs in size. So you also want to have ways of separating things that are hundreds to thousands of base pairs in length. And that's normally done with agarose gel electrophoresis, much bigger holes, bigger pieces of DNA that can go through it. This, unfortunately, doesn't have a size marker down here, but this is a couple of hundred nucleotides. That's about 1,000 nucleotides, getting up to about 10,000 nucleotides here as well. That's great if you've chopped up your particular piece of DNA, but if you're trying to look at individual chromosomes, tens of thousands is really kind of small. We're talking millions usually. So there's a neat technique that people came up with, again, probably about 20 years ago now, called pulsed field gel electrophoresis. And what this is instead of having a negative charge at one end, your positive charge at the other end, and everything going at the same rate through the same gel, is every relatively short period of time, you switch the polarity that you have on your gel. And what that does is it takes your nucleic acid, it unfolds it, tries to go in the opposite direction, then has to unfold and refold again. And the bigger the piece of DNA you have, the longer it takes to do that. And so this technique allows you to separate literally millions of base pair long DNAs by this changing your electric field multiple different times. So this is great. We can separate single nucleotides. We can separate millions of nucleotides relative to each other. But how do we see the DNA? Well, classic way to see the DNA is to take our DNA. This is what's usually done with our agarose gels. By the way, these pulse field gels are also using agarose with the large pore sizes. And then stain that DNA with something that makes it visible. And the best examples of those are what are called intercalating dyes. So that's what's used here. Ethidium bromide's the best known of those. But ethidium bromide and most of these other intercalating dyes have zero fluorescence when they're not associated with DNA. But as soon as they get into your double-stranded DNA, then they fluoresce. And so that's these fluorescent dyes, ethidium bromide, classic mechanism to use for looking at these agarose gels. Acrylamide gels, one of the problems here, as soon as you think about doing intercalation, is intercalation is actually going to change the size of your DNA. So if you change the size of your DNA and you have single nucleotide resolution on a particular gel, this is going to be a real problem. And so here, you almost always use radioactivity or some other mechanism for actually detecting these bases rather than an extra stain which you can put into it. So how do you do this detections? Usually it's by hybridization, and we've talked about hybridization since way back when, the very beginning of the course. Any kind of nucleic acid can hybridize. DNA can hybridize to DNA. RNA can hybridize to DNA. RNA can hybridize to RNA. And the key here is you have a short piece of whatever nucleic acid it is. It turns out it's easier to make DNA that has some kind of label on it. So that's the red stuff here. And again, literally, you can go to websites and type in your favorite nucleic acid sequence, and you will get sent that particular nucleic acid sequence. Then you want to have a label on all of these guys that are A's, and what you will find, as long as you do your hybridization under the correct conditions, that the only thing which is going to bind to your favorite sequence here out of the mixture is the one which is specifically hybridizing to it. So yes, you can get these DNAs. We're not going to get into DNA synthesis. Wonderful class next term on nucleic acids, Biochemistry 492. Have a chance to take it. Dr. Lehman's fabulous. Talk all about that stuff. Yeah?
You said it's easier to make DNA if it's what?
So it's just easier to make DNA than RNA chemically. And again, we can talk about that all next term again in Dr. Lehman's class. But once you've made it, it's still not easy to detect that very specific piece of DNA. So usually people, well, in the old days, again, back when I was in graduate school, used radioactivity for labeling. Now people usually use fluorescent tags instead. Still, the same idea, however, is that you have to have some way of detecting that particular piece of DNA. So there are really two different ways of doing that. We'll start actually down here with the bottom one, end labeling. So end labeling is literally labeling the end of your DNA. What ends do you have on a DNA? 5' and 3', right? So two different ways that you can do that. At the 5' end, polynucleotide kinase. What do kinases do? They'll phosphorylate things. What's it phosphorylating? Any kind of nucleotide, a particularly polynucleotide. So it's really good at putting phosphates on the end. And if it's a radioactive phosphate, here are yellow one, then your radioactive phosphate will get put onto the end of your DNA. Terminal transferase does exactly the same thing at the 3' end. It just exchanges off that last nucleotide. So now you've got a piece of DNA that's got labels at the 5' ends. Now, this is great, but you usually want to just have one of those strands that you're interested in binding to and using it as a probe to detect something. You don't want to find the complement of that. You want to just find that one piece. And so usually what you do is you'll cut off one of the ends, like you have here, and just keep this one piece that has a label on it. So that's one way of doing labeling. The other way of doing labeling is taking advantage of everything that we all know, right, from DNA polymerization. We're really good at DNA polymerization. Yes, we had it at the last midterm, so who cares? But DNA polymerization is a great way to add radioactive nucleotides that are complementary to a particular sequence that you're looking at. So if you have little primers, these orange bits, then you can put in DNA polymerase and radioactive or non-radioactively labeled nucleotides. It will extend and will give you a piece of labeled DNA. You can do this with specific primers, which again you can buy from your favorite nucleotide supply store. Or what's very often done is taking random primers, also called hexamer primers. And so this is a whole mixture of different primers that'll bind all over wherever your particular piece of DNA is. Another really useful tool in terms of making these kinds of labeled intermediates is using DNA polymerase 1. Why would you want to use DNA polymerase 1? What's the really fascinating part about DNA polymerase 1 that none of the other polymerases have? It was so long ago, Dr. Stedman, I can't remember. So it has both proofreading exonuclease activity, but much more importantly, 5' to 3' exonuclease activity. So if you have DNA polymerase 1, you don't have to pull these two pieces of DNA apart before you can have all of your annealing, primer extension, etc. If you just have a tiny little break in the DNA, like we have on our model here, then DNA polymerase 1 can bind to that and then replicate its way along because it's got this 5' to 3' exonuclease activity. And so they're actually giving you a labeled piece of DNA. Again, old school, old guys like us like to use radioactivity. Radioactivity is wonderful. Talk much more about it later. Some people don't like radioactivity. So more and more people are using what we call non-radioactive labels. Turns out that there are lots of DNA polymerases that can add nucleotides that have extra labels on the end of them. And the DNA polymerases do a fine job with this. These are labels that are hanging out in the major groove. That particular piece of DNA is not going to be really good for having proteins bind to it, but it's going to be fluorescent or whatever other kind of probe you want to have here. So this is just one example of these modified nucleosides. Here, nucleoside triphosphate with a almost normal base attached to it, and then this extra spacer. And DNA polymerases do just fine with this. They put in exactly the right nucleotide under these conditions, but now you have a label hanging off, or I say an extra piece of chemical hanging off the edge of this. Often, again, this will be something which is fluorescent. In this case, it's a molecule which biotin binds really well to. Biotin is, let's say not biotin, it's a antibody which binds to deoxygenin, makes it really easy to detect this particular piece of DNA. So why are we doing all this work in terms of finding all these probes? Why do we care about all of these extra pieces? Why do we have to be able to detect them all? Well, this has to do with finding the needle in the haystack. And that's really what most of these techniques are about, is finding that needle in the haystack. The one piece of DNA in the 3 billion base pairs of our genome that's the interesting one, the one that we really care about. So how do we do that? We have a little piece of DNA which we've now labeled, either end labeled or labeling through DNA polymerization. And now we want to find where that piece of DNA is and separate it out from all the rest of the DNA. So this is a technique called blotting, and the probe is what you're using to try and detect that. What you do is you have a gel here with all of your RNA, DNA, etc. separated on it. It's going to be a whole mixture of different DNAs, different RNAs. In the gel, it's actually kind of hard to detect, so you build what we always used to call sandwiches in the lab to transfer this nucleic acid onto a piece of paper. Turns out it's a piece of paper that binds really well to nucleotides. And now we have a copy of what was on this gel on this piece of paper. And it's nice because it does two things. One, it concentrates all of the nucleic acid that was on the gel. And second, makes it available to something that you put down on the outside. What is that something? It's your labeled probe. And then if you've got radioactivity or fluorescence, you can now detect where your favorite nucleotide happens to be just by looking at where we have them down here on our blot afterwards, detected with autoradiography, just putting a piece of film on top of it. Or in these cases, we've got scanners that will detect the radioactivity or detect the fluorescence. Multiple different kinds of blots. We talked last time about protein blots. Those are called Western blots. Why Western blots? We'll get to that in just a second. The original kind of blot that people used was something called the Southern blot. Southern blot is DNA that you separate on a gel and you hybridize DNA to it. Now, why is it called a Southern blot? Because a guy by the name of E.M. Southern came up with it in the first place. If, however, you're separating RNA, it's called a Northern blot. Why is it called a Northern blot? Because you already had Southern blots. Why Western blots? Because you already had Northerns and Southerns.
What is the northern blot?
So Northern blot is RNA. So you separate RNA, and then you hybridize to a particular RNA. So that's the process. Again, to be able to detect how big any of your pieces are, you want to have markers of a particular known size. There are lots of ways of looking at these markers. But again, the important thing here is detecting of each of these individual bands. Southerns, we're looking for specific DNAs. Northerns, we're looking for specific RNAs. And Westerns, we're looking for specific proteins. Easterns actually haven't been developed yet. I know people have tried to think about what other kinds of blots they would use, but DNA, RNA, and protein, what else would you want? tRNAs. Well, RNAs are usually going to be northerns then. So, but people have talked about these different things. But it turns out that's actually a great lead into the clicker question, which is on the next slide. Which is here. To detect a DNA binding protein using a blot, you should consider a northern blot, southern blot, western blot, northwestern blot, or a southwestern blot. Ten, 5, so I heard somebody mention that I was aiming for 100%. That's true, I was aiming for 100%. But unfortunately, this is a 0. It's protein and DNA. So it's a southwestern blot. Now, since I normalize, it makes no difference. If nobody gets it, then nobody gets points and it disappears. There are also what people call far western blots, where you look at protein-protein interactions. There's all kinds of variability in this. Yeah? Pardon? So the question is, how do you do a southwestern blot, basically? So what you do is you do a western blot where you separate proteins, but then you detect it with DNA, the DNA that's going to bind to that protein. Yeah. So the question is, instead of putting antibodies into your western blot, you put in DNA. Exactly. Yeah. Right. So the western blot, which is the protein. So western blot is protein, and then the DNA is the southern blot, so it's a southwestern blot. So if we had an RNA binding protein, what would it be? Northwestern blot. Yes, exactly. Mm-hmm. But the best answer would be a southwestern blot. But again, nobody got it, so it gets normalized out. Obviously, it was a bad question, right? Actually, it wasn't meant to be a bad question, believe it or not. So that was all about blots. Now, why do you care about finding this one particular piece of DNA? And you've gotten your DNA, you've chopped it up, you've done your western blot or your southwestern blot, your southern blot. You know what size that one particular piece of DNA is. Well, how are you going to work with that one particular piece of DNA? You know it's there, it's in this big mixture. Well, one of the ways to do it is to use what are called libraries. And so what's a library? A library is basically a collection of all the pieces of DNA in a genome. And literally smaller pieces, because you can't deal with the individual chromosomes. You want to have smaller pieces of your DNA. And so the way that this is done is you take your favorite genome, again, usually human genome, a bacterial genome, etc., chop it up at random, and then put it in a place, and we'll talk more about these plasmid vectors in just a second here, which can make many, many copies of that one particular piece of DNA. But of course, you've got all the different pieces of DNA. So you have to separate each of these different pieces of DNA into different bacteria. So one bacteria's got one DNA in it, the next bacteria's got a different piece of DNA, and so on and so forth. And then each of these bacteria here can make more copies of themselves and make many, many copies of that plasmid DNA with an extra piece of DNA in it. Now what you have is a whole bunch of bacteria, each of which is a slightly different piece of DNA in it. Now you want to find out which of those bacteria have the DNA that you're interested in them. How would you do that? Use your probe, which you made before, to hybridize now to that particular bacterium. And so that will be able to identify which of them has your favorite piece of DNA in it. So this is great, works actually extremely well if you're looking at bacteria, but if you're interested in a particular protein or a protein coding gene, what happens to the vast majority of protein coding genes in our genome? They get spliced. So what does that mean about just chopping up the genome into pieces like this and finding a piece of DNA that has the piece of DNA that you're interested in it? It's got all of the extra bits. So it'd be really nice if there were a way to just look at the RNA, which is being made after all of the processing, and then pull out those things. And that turns out to be what's called a cDNA library or a complementary DNA library. This now takes processed RNA from a particular cell, and then with that processed RNA makes a DNA copy of it. How do you make a DNA copy of RNA? Reverse transcriptase, exactly. So reverse transcriptase, what kind of primer do you want to use for the reverse transcriptase? Poly-T, because that will now hybridize to the poly-A tail at the end of your processed messenger RNA. That then gets extended with your reverse transcriptase. Reverse transcriptases often have RNase H's associated with them. What does an RNase H do? Chops up the RNA in an RNA-DNA hybrid. And then you can extend, this is either the reverse transcriptase or a DNA polymerase, which will extend this DNA and make an extra DNA copy. There are a couple of problems, however, with cDNA libraries. And the main one is that just like with telomeres, there's always going to be a little piece at the 5' end which is missing. Because you have to have a primer, that primer gets degraded. But on the other hand, it's a really useful way of getting all of your coding sequences, and particularly this is going to be a snapshot of what genes are being transcribed and those messenger RNAs are being processed in terms of what's being made at any particular time. So two different kinds of libraries. If you care about what kind of splicing could happen and how splicing is taking place, then you definitely want to have a genomic library because that's looking at just the DNA, which you started with. If, on the other hand, you're just interested in what a particular cell at a particular time is making in terms of its RNA, then you want to have a cDNA library. But this cDNA library is just going to be from those genes which are being expressed. Any genes which are not being expressed, you're not going to see. If they're expressed at very low levels, it may be very hard to find them in your cDNA library. You may have to have them in your genomic library. So nice things about these libraries, particularly for things like humans, they're actually kind of a pain to make yourself, but they're incredibly useful for researchers who are looking at them. So again, just like those little pieces of DNA which you can order libraries. And there are a number of companies which have cDNA libraries from brain, cDNA libraries from liver, cDNA libraries from cancer cells, etc. And so you can just buy most of these libraries instead of having to make them yourself, and those libraries have much better quality control than you would be able to make in the lab yourself. So now you've got a library, you've used your probe to find which particular piece of DNA in that library you're interested in. Now you need a lot of it, or you want to make the protein which is coded for by that particular cDNA. So how do you do that? Now we do recombinant DNA. Remember all those restriction endonucleases we were talking about? You take that piece of DNA, cut it with a restriction endonuclease. We'll take a vector or another piece of DNA, cut it with either the same restriction endonuclease or just a restriction endonuclease that gives you the same overhang at the end, the same sticky end. So here, TTAA can only, actually say AATT, can only hybridize to AATT, can't bind to any of the other ones. This process of ends coming together people also call annealing. Hybridization, annealing, they're just two different terms for the same thing. Base pairs coming together. Once you have this base pairing, you still have little breaks in the DNA on either side. You can now put these two together, and lo and behold, you have DNA, red and black in this case, or two different sources of the DNA that you started out with. And so here we have a recombinant DNA molecule. And people were really scared about all this, genetic engineering stuff, literally in the late 70s when they first came up with this. The first example, of course, was using viruses, putting stuff together. So they had a big meeting called the Asilomar meeting, at which point they said, okay, we're going to have a moratorium on doing any of this stuff until we think about some safety characteristics, which was really, they had scientists, ethicists, politicians, et cetera, got together, talked. Wow, talking in politics, what an idea. But then came up with lots of rules and regulations in terms of how to go about doing these kinds of technologies. And part of that is every institution, like Portland State, has an institutional biosafety committee whose charge is to look at any experiments that are happening with this. You're looking at the chair of that committee. So these are the kinds of things that people were very interested in then, and we've got a whole set of regulations. Basically, reduce risk as much as possible. And it turns out the risk is pretty minimal relative to this recombinant DNA, unless we're talking about Ebola kinds of immune suppression activities, which none of that happens here, at least not that I know of. So why do most people do this kind of recombinant DNA technology? It's mostly to try to get many copies of one particular piece of DNA. So how do you do that? You have your piece that you've gotten from a library, or you've detected it somehow with your southern blots, and then you want to take that piece and make many, many copies of it. How do you do that? It's usually through what are called plasmid vectors. Plasmid vectors are small circular DNAs that were originally found in lots of different bacteria. Turns out that they're really good at spreading antibiotic resistance, which is why people were interested in them in the first place. But then a couple of smart people said, hey, we could maybe use these to make lots and lots of our favorite piece of DNA. So the way that you do this, you have one of these plasmids. You cut it with your favorite restriction endonuclease. You've got your piece of DNA that you want to make many copies of. You've cut these either with the same restriction endonuclease or those that have sticky ends that work with each other, and then put them together. You've got your recombinant DNA. This now you can make many, many copies of. This is actually a classic kind of vector down here at the bottom, pulled off of a website here. Got a couple of things that are important on it. One is this origin of replication. And the PBR 322 is literally from Paul Boyer and Roberts, who are the people who started this thing again in the late 1970s. Origin of replication. Origin of replication, also an antibiotic resistance gene. Yes, this is how they're originally found in the first place, but when you do this recombinant DNA experiment, you've got to take that DNA and now put it into a bacterium. And you need to know that that bacterium has got that extra piece of DNA in it. So how do you do that? Antibiotic. You treat with antibiotics. Only the bacteria that have this extra DNA in them will be resistant to the antibiotics. All the other bacteria are going to die. Yeah?
Is there a risk of only part of the vector being integrated? Like, say, that you only got the antibiotic resistance part?
So the question is there a problem with just getting only the antibiotic resistance part as opposed to your favorite gene, which goes in there? Yes, it turns out to be a huge problem. You can talk to people in my lab about all of the cloning hell, we call it, with ending up with a wonderful plasmid that doesn't have the piece that you're looking for in it. There are ways of dealing with that, but that's a subject for the recombinant DNA techniques theory class next term, which Dr. Bartlett will be teaching. Also in this plasmid, we've got lots of restriction endonuclease sites, also called a multiple cloning site. So this is where you cut your piece of DNA and put it into. This particular one also has a tag conveniently put in there, so you can use that for purification. You can use it for labeling inside your cell. And then also has a promoter here called the T7 lac promoter. Well, remember the lac promoter we talked about in terms of DNA regulation in E. coli? What regulates it? The lac repressor. So we've got the lac repressor on our plasmid as well. That's what this guy is. This is your gene regulatory protein. And then this particular promoter, which is right here. Turns out that it's also associated with viruses, and we'll talk a bit more about that as well. But here, origin of replication, antibiotic resistance, and place that you can put your favorite piece of DNA. So once you've done that, you've got your DNA, you put it into the cell. Now we have antibiotics. Now we can grow this particular bacterium, make many, many, many of them. And now you've got many copies of your purified plasmid, which are here. You can also, instead of purifying your plasmid, you can also have, again, if you've got the appropriate promoter and regulator, you can make a lot of RNA, and that can actually get turned into protein. So you can make lots of protein as well as making, excuse me, your DNA. What a lot of times people just want the DNA for, if you're trying to do DNA sequencing, it's also very useful to have lots of copies of one particular piece of DNA to use for DNA sequencing. And one of the very useful things, particularly in terms of the human genome, is a very specific kind of plasmid here, which is called a bacterial artificial chromosome. It's basically just like the plasmid we looked at before, only you can take really, really big extra pieces of DNA, literally hundreds of thousands of nucleotides in there. And so instead of having a plasmid origin of replication that we looked at on the last plasmid, it actually has the E. coli origin of replication, or EC, which is in there, and that can replicate then very large pieces of DNA. But if you want to make protein, we have our promoter sequence that we looked at just on that last slide. In some cases, again, there'll be a flag, or say a tag right next to it. Flag tag is one of them. Now you cut with your favorite restriction endonuclease, put in your sequence here, and then take this, put it into cells, and instead of purifying the DNA, now it'll be transcribed and made into protein. And so here's a nice example of that. Here's your bacterial cell that has the plasmid in it. And then as you change the temperature, this particular promoter is a heat shock dependent promoter. Then you start making more and more of your favorite protein, which is right here. Yeah?
So going back to the back,
Mm-hmm. Yeah.
when we talked about bacterial evolution, we talked about bacteria likely keeping or having their chromosomes kept at a small size because otherwise they wouldn't have been able to be quite as prolific. Am I remembering that correctly?
Okay, so this question is really much of an evolution of bacteria question, probably best talked about after class. So the idea here is that you can put in tens of thousands and hundreds of thousands of nucleotides, not millions of nucleotides, so not making it way bigger than your genome otherwise would be. So here, again, it's just a nice example. This is STS page of, you can see all of the other bands on here all represent the E. coli proteins, and now you're making more and more of your favorite protein here. If it happens to have a tag on it, then that's a really nice way of purifying that protein, so you don't need to go through all the steps that we talked about last time, just an affinity purification, or detecting it inside your particular cell with some kind of antibody to that tag. So cloning with libraries and hybridization and taking those, putting into cells, growing a whole bunch of it is great, it's wonderful, but practically nobody does it anymore. Why? Because of a really crazy guy, and literally crazy. If you want to see the most bizarre Nobel Prize speech ever, go and look up Kary Mullis. But he came up with this technique called the polymerase chain reaction. And the polymerase chain reaction is, instead of taking all of your DNAs and putting them into E. coli and then making many, many copies in your E. coli, ligating, doing all these extra steps, if you just do the polymerase chain reaction, you can make many, many copies of a specific piece of DNA without going through all of those extra steps. So the way that the polymerase chain reaction works, as most of you have heard this before, double-stranded DNA, heat it up, put in primers of your DNA, specific sequences. So we've got genome sequences for practically everything now. You can get these primers. These primers are going to anneal to opposite strands. You amplify in the presence of a normal DNA polymerase, but a DNA polymerase which is stable at high temperatures. This was the trick that Kary Mullis didn't come up with. These thermostable DNA polymerases, these will extend from those primers, and what will happen is these will extend well beyond, and it has to be beyond where your primer on the other strand was. Now you can separate these strands again just by heating them up, put in new primers, those can extend, and over repeated rounds of this, you'll end up with more and more, an exponentially increasing amount of just the piece of DNA that you're looking at and these primers, which are at the end. And one of the real tricks about doing PCR is you can add extra bits to the 5' end of your primers. So you can put basically whatever tag you want onto your PCR fragment just by adding a little extra piece to your primers, which would be what's on the clicker question, which we're going to skip for time reasons today. So no one's going to get any clicker points today. How sad. Everyone gets 100%. Perfect, yes. 100% is 0. Wait a minute. Okay. No, but because I really did want to talk about DNA sequencing, because this is a very important aspect of the course. And we'll just finish up talking about DNA sequencing here. Couple of different kinds of DNA sequencing. The classic DNA sequencing is called dideoxy sequencing. Why is it called dideoxy sequencing? Because we've got our base here covered up by our clickers, which has no OH at the 3' position. If you have no OH at the 3' position, then whenever you're polymerizing and you have some of these dideoxynucleotides, you can't extend that particular piece. So all you need to do is have a sequencing reaction which has small amounts of these dideoxynucleotides in that reaction. And your DNA polymerase will come along and occasionally will incorporate one of these dideoxynucleotides and can't go beyond that. if you have a whole mixture of DNAs and 4 different reactions, each of which has a different dideoxyribonucleotide in it, you'll end up with a whole mixture of DNAs. Some that stop with A's at different positions, some that stop with T's at different positions, some that stop with C's at different positions, and some that stop with G's at different positions. 4 different reactions. Each of these have slightly different lengths. Those lengths are going to be stopped by wherever you had your dideoxynucleotide. Now, if you take a high-resolution gel, so that's that acrylamide gel that we talked about right at the beginning, separate these based on each of the reactions, your dideoxy A reaction, your dideoxy T reaction, your dideoxy C reaction, your dideoxy G reaction, and then look at each of these pieces. Since it's single nucleotide resolution, you can literally read off the sequence here. This is A, T, G, T, C, A, G, etc. Just from the bottom to the top, because the smaller runs at the bottom, the larger is going to run at the top. So this is a way that you can do sequencing, and this is the classic way that sequencing was done for many years, even, again, back in the dark ages when I was in graduate school. A couple of improvements happened, actually literally while I was in graduate school. Some of these things happened. And the main thing here was just taking those 4 different reactions. So we had DDA, DDT, DDG, and DDC, and squishing them all together into one part of your gel. And so that's basically just shown here. Each of these now, instead of all being the same color, and that color was almost always radioactivity, now they're different colors because you have a different fluorescent dye on each of these dideoxynucleotides. And what that means is you can put all of these now into one well of your gel and separate from the bottom C, A, T, etc. The way this is also done is with an automated sequencer. Literally, it's a laser, actually a set of lasers here, which will illuminate whatever's coming off the bottom of your gel, and each of these peaks will represent one of those dideoxy-terminated pieces of DNA. And any of you who look at sequences next term in the Mutant Viruses from Hell lab, we'll look at sequences. We'll actually get these chromatograms back, so sign up. Still a few more places left. These are the kinds of sequences that people get. You can do about 700 nucleotides, in some cases up to about 1,000 nucleotides, using this kind of technique. This is really nice because these capillaries, so instead of being a gel, a big flat wide thing, you can now run all of these gels in single capillaries. And the great thing about single capillaries is you can do a whole bunch of them at the same time. And this is the fridge that made the human genome possible. It's basically taking that technology and doing 96 sequencing reactions at the same time and running them through these machines basically 24/7. All the details on this are not really critical, but it was really sort of this technology which allowed the human genome sequence to be done. So it's a specific DNA sequencer, and again, kind of looks like this. At that point, it was doing 96 reactions at the same time. Now I think they're up to 512 reactions at the same time. Then it was about 500 base pairs per run. But basically, 14 of these could do a human genome in 1 year. Now, I just told you right at the beginning, right? We've got these sequencers that can do 6 trillion bases in 2 days. So it's gone up a lot. Human Genome Project took about 10 years and about $3 billion to do. Now, you do all these sequences, and I said, you've got about, on a really good day, 1,000 nucleotides. How do you take 1,000 nucleotides and make a 3 billion base pair genome? So there are lots of different ways you can do it. You can do 1,000, from 1 to 1,000, from 1,000 to 2,000, 2,000 to 3,000, etc. That would take a ridiculously long time to finish the genome. This is also called genome walking, because you start from one, you go to the next one, go to the next one. What, again, was really critical in terms of understanding the Human Genome Project was actually progress in computers, because the way that they did the human genome sequence was what's called a shotgun sequence. Basically take all of those sequences, break them into smaller pieces, 1,000 bases each, and sequence every single one of them. And then, using a computer, find the bits that overlap with each other. Because if you've got a random piece of DNA that's been broken into smaller pieces, if you do these 1,000 base sequences, find out where they overlap, you can piece back together the jigsaw puzzle that you started with. And so that was the real computational breakthrough, as well as the technology of doing these sequences. Computers are also really important for gene prediction and comparative genomics, but here, main thing is talking about how you can take smaller pieces and then assemble them all back together with each other. And this gets us to those bacterial artificial chromosomes that I was talking about before. Bacterial artificial chromosomes are hundreds of thousands of nucleotides in length. You can't sequence each of those individually, but it turns out that given the computational power that they had when they started the Human Genome Project, you couldn't take 3 billion base pairs and piece all the different things together. What you had to do was take smaller pieces, which turned out to be exactly the size of the bacterial artificial chromosomes, and use those pieces to fit together. And so this is an example here of one human chromosome. You can make a bacterial artificial chromosome of each of these different size pieces and find that they overlap with each other. This overlap you can find either by just sequencing, which is what we do now. At the time, it was where individual restriction endonucleases bound. And so what happened with the Human Genome Project is they made a big library of all of these different back clones and then sent each back clone to a different place that did sequencing. They chopped it into much smaller sequences and did the sequencing there, then sent all of them back to the headquarters, as it were, of the Human Genome Project, and they did sequencing of putting all those sequences back together. And that's how we got the whole Genome Project together. Next generation sequencing is the latest, and I just wanted to go over this really quickly. This is sort of the standard now for next generation sequencing. I hate next generation sequencing. I think multiply parallel sequencing is a lot better. Here, instead of having those little capillaries where you're doing your sequencing reactions, now it's just done as little dots on a microscope slide. And literally billions of dots. And you add nucleotides here. Each of them has a fluorescent dye. And literally what color lights up here is what nucleotide has just been added at that one particular spot. So if you've got a microscope that can scan all of these things really quickly, you can literally get to these kinds of insanely large numbers, 6 terabase pairs in 2 days. So that's all for material this term. Thanks for hanging in there. We'll have a review on Friday, exam on Wednesday.